Hi, and welcome to this session. My name is Liz Mansfield, and I'm the Deputy Office Director for Personalized Medicine in the Office of In Vitro Diagnostics and Radiological Health. Today, I'm going to discuss the development and use of reference standards materials and datasets for NGS and how FDA might use them in a regulatory context. You've seen the graphic on this slide before, and I hope you've read the paper that it was published in. It describes the areas identified as necessary for success in the roadmap for regulatory science in NGS informatics. I'm going to focus on the specific issue of creating gold standard reference data sets, which will be critical for understanding the performance of many NGS tests that are developed, whether they're used in research or for clinical applications. I will also discuss a little about how FDA might use these data sets in a regulatory setting and the potential for evaluating a test by comparison of test results using Precision FDA. Let's start by posing the question of what truth you might compare to in order to know that your test has correctly identified existing variants across a given genome. We will assume here that you're developing a test that is not necessarily targeted to detect pre-specified variants, although you would have similar issues for a targeted test, or even for a test that only detected one or several known variants. In the non-targeted case, you will need to know how well your test performs in detecting the correct bases over wide areas of the genome. You won't generally need to know that your test can detect every possible variant, because that would be nearly impossible. In next-generation sequencing and some other molecular technologies, each component of a test has to work in a manner that allows for accurate results. The library reagents must correctly select and amplify, if needed, the targets of interest. The instrument and its associated chemistry must correctly detect which base is at each position in a fragment. And the informatics must correctly assess the sequence quality and assemble the fragments correctly into a linear sequence. Comparison of the sequence is generally to a reference sequence, often the human genome build, and variants must be detected. Since every person will generally have a unique set of variants, it's nearly impossible to know exactly what variants will be found. And this makes knowing what truth is more difficult. So how do we get to the best possible accuracy in determining sequence and variants when we don't know what variants will be found? Ideally, genomes would be simple and uniform, and the NGS system would be perfect at sequencing and detecting variants. To know this, ideally, we would have a reference material for every possible variant that could be detected. However, with billions of bases, or some subset of that, this isn't realistic. Genomes are complex and highly variable, and clinically, we generally aren't interested in normals. To date, test systems aren't perfect and aren't expected to be so for all genomes. It also seems unlikely that we will ever know all the variants that exist in the human population, and reference materials are necessarily relatively scarce. The reference materials that we use need to be uniform and well studied so that we are confident in their sequences, and they should contain the types of variants we expect to encounter in both normal and diseased genomes. To be able to generate a stream of useful reference materials, we need to determine the characteristics needed to make a reference material useful, so that we can identify candidate materials and appropriately characterize them as sources of truth. Several groups around the world have been thinking about reference material characteristics. The Genome in a Bottle Consortium here in the US considered that a good reference material would need to be available in sufficient overall volume, of course, then aliquoted into individually useful volumes, to last over many users and uses. The reference material needs to be uniform so that there aren't many internal variables in the sequence. Reference materials need to be renewable so that they can be replenished as aliquots are used up. They need to be broadly available to whoever needs them, and they are only useful as reference materials if we have confidence in the data set or sequence that is established as the true sequence. They also need to have all or most of the types of variants that are expected to be encountered in research or clinical sequencing, so that developers can have confidence in their ability to call all types of variants in different genomic contexts. Ideally, we would have multiple reference materials that represent population-based genomic variability across many different populations and contain known pathologic variants to aid in specific variant validation. This sounds like a lot of requirements, but realize that not every reference material has to have all of the necessary characteristics. We can also use several materials together to establish confidence, with each material providing a different set of variables of interest. An example of a genomic reference material is the very first human genomic reference material produced by the National Institute of Standards and Technology, also known as NIST, which is available now, and the genomic material and dataset have already been widely used. NIST will be releasing additional materials over time and has released some additional datasets already. Through the Genome in a Bottle Consortium activities, we learned that development of reference materials is very much aided by a community process, which provides multiple inputs from different test systems, informatics approaches, and methods to resolve differences in variant identification. We think that one type of model is cost-efficient and relies on a type of crowdsourcing where everyone contributes. This helps us to more quickly develop materials that respond to clinical and research needs and drives improvement in data sets over time as we learn more about why certain genomic sequences are hard to establish and get more and more data sets that help to correct errors in earlier iterations. This model can also start as a private effort that is then open to the community for input. To date, there are 3 NIST reference data sets available for use, and I encourage you to go to the NIST website to access these. I believe it's also available on the Stanford website. And look and see what's there. Certain privately generated data sets have also been released to the public, and some of these can be found on Precision FDA, where they've been deposited for use. Next, we need to consider which types of data sets are most needed, identify appropriate materials, and get started on generating them following a general process of identifying materials, getting users to run them on their system configurations, and perhaps using Precision FDA as a repository for such sequences, where NIST or some other body can integrate the data to create a truth set with confidence figures, which would improve over time as more data is deposited and errors are detected and fixed. How will all of this help in regulatory science? FDA has already partially established the process in which an applicant provides, in a regulatory submission, data demonstrating that the test system in question can generate correct calls over several reference type genomes, spanning the variant types in multiple genomic contexts. Right now, this is through a standard submission process to FDA where the static data are sent for review. We envision a future where FDA might be able to perform an interactive review, perhaps using Precision FDA, where data sets to be analyzed can be selected and specific performance demonstrated. We don't have the capability now, but it would be a goal worth thinking about. We also need to consider the best way to regulate an entire test versus regulation of components, since different developers will want to combine different components to suit their needs. One example of this is FDA's clearance of the Illumina MiSeq DX instrument and reagents. For more information on the questions asked and the studies provided, please take a look at the decision summary online. Here's the URL. Thank you for attending this session. I hope you learned something new and will think about reference materials more as you complete this course.